Hello biology students, and welcome to video 7 of the organic chemistry unit, where we're going to be talking all about enzymes. So remember, in last video, in video 6, we talked about a whole different host of functions of proteins, and one of those functions was that proteins can be enzymes. So enzymes, the function of an enzyme is a protein that speeds up a chemical reaction by decreasing the activation energy. Okay, so basically enzymes are going to make reactions faster by decreasing this thing called the activation energy, and we'll talk about that in a little bit. Enzymes also do not change. So this means the same enzyme can speed up a chemical reaction, and in that chemical reaction the reactants are turned into products, but the enzyme remains unchanged, and then the enzyme could go ahead and do another reaction and another reaction. In other words, they're not used up. They can be used over and over again. So in the last slide, we said that enzymes work by lowering activation energy. Well, to understand how they work, we have to understand what activation energy is. So what activation energy is the energy required to start a reaction. Basically, the way it works is for a reaction to start, for a reaction to begin, the reactants, the things that are going to react together, have to absorb energy, okay? So you have to put energy into a reaction to get it started. And the reason for this is if this wasn't the case, molecules could just spontaneously start reacting by themselves. And that would be bad if the molecules on our planet were just spontaneously changing into other things. We want to have a way. We say, okay, if we're required to put energy in, things won't just spontaneously break down. So if we look at a little picture here, normally I have reactants. So these are the molecules that are going to react together to form some kind of product, okay? And in order for these reactants to become products, they have to get enough energy to get over this activation energy barrier. And so kind of the way an enzyme works, okay, and remember this is just a schematic, this is not actually what's happening, is what an enzyme does is it lowers this activation energy, so it basically makes it easier for reactants to turn into products. So by definition, an enzyme is something that's decreasing an energy barrier. And by the way, another word for an enzyme is a biological catalyst. Okay, so just keep that in mind for when you see that vocabulary word. So here's another graph that shows the same thing. So you can see on this axis I have energy, okay, and on this axis I have progressive reaction. So in other words, the beginning of the reaction's over here, the end of the reaction's over here. So you can see normally, if I do not have an enzyme, I have to put a bunch of energy into my reaction in order to turn from reactants to products. However, if I have an enzyme, okay, I have to put a lot less energy in for the reaction to occur. So how do enzymes work? How do they actually do this? So let's look at some vocabulary first. So first, the first vocabulary word I want you to note is the word substrate. So this is the reactant, this is the molecule that the enzyme is going to work on, okay? The active site is a particular place on the enzyme, so it's a part of the enzyme where the substrate is going to bind. And basically the way enzymes work is they position substrates in such a way so the reaction happens faster. In other words, they kind of put them in the correct orientation for optimal reaction. So let's look at this in pictorial form. So please ignore this giant black blob. I was trying to edit the slide and I was having some issues, so I just blacked something out. Okay, so here's my enzyme. And in particular, this is the enzyme sucrase. It's the enzyme that breaks sucrose, our disaccharide, down into its monomers. Okay, so here's the active site. This is the spot on the enzyme where the substrate is going to bind. And then our substrate is sucrose, because that's the thing that's being broken down. So what happens if you look, sucrose, the substrate, binds to the enzyme in the active site, and then the enzyme positions sucrose in such a way that it's really easy for water to come in and via hydrolysis break that bond down into glucose and fructose. And if you look, my enzyme remained unchanged, and in theory it could go bind to another sucrose molecule to continue the process. Okay, so a couple things about enzymes. They're specific, which means there's a different enzyme for every chemical reaction that occurs in your body, which is pretty crazy. So there are really thousands and thousands, maybe even millions of different enzymes in your body. So we say that only one enzyme can catalyze or react to do one particular reaction. Okay? And this is because every enzyme is going to have a different active site, which means it's able to bind to a different substrate. So this is true because enzymes are specific because their shape only fits one substrate. Okay? So back in the several years ago, it was thought that enzymes and substrates fit together like a lock and a key. And so it's called the lock and key theory, was that each substrate fit exactly into the active site. However, now we know that's not quite the case. So instead, what's true is what's called the induced fit model. And this says that enzymes, their active site generally fits the shape of the substrate. And when the substrate binds, what happens is the enzyme actually changes shape so that way it will fit perfectly. So it's the induced fit. It's induced or kind of forced to change when the substrate binds. So enzymes are important for a lot of reasons. They basically do all of the work in your body. If you didn't have enzymes, there was no way you'd be alive. So enzymes regulate all your chemical pathways. They synthesize or build all the materials needed by your cells. So they do all the dehydration synthesis reactions. They catalyze all the reactions that release energy. Okay, so you wouldn't have to be able to make any energy without enzymes. They regulate all the reactions for information transfer. And basically, because enzymes speed up chemical reactions, if you didn't have them, most metabolic reactions would occur too slowly to sustain life. So a reaction that happens in a millisecond with an enzyme would probably take hundreds of years to occur on its own without an enzyme. Okay, so enzymes are so important that it's really important that we maintain stable body conditions because enzymes are influenced by many different things. They're influenced by temperature, so we have to keep temperature constant in our body. They're influenced by pH, concentration of substrate, and a whole bunch of other things as well. So let's look a little bit at these. So temperature first. So it turns out that the rate of a reaction, or how quick a reaction happens, it's pretty dependent on temperature. Okay? So if you think about it, at warmer temperatures, molecules are moving faster. And if molecules are moving faster, you get more collisions between the enzyme and substrate. So you can kind of think, when it's cold, because molecules are moving really slowly, it's not likely that the enzyme and substrate are going to collide, so the reaction doesn't happen very quickly. But as it gets hotter and hotter, molecules are moving more quickly, things are colliding, and therefore the enzyme and substrate react together more often. However, at some point you hit a peak, and this is where you get too hot, and you actually denature your enzyme. You denature the protein falls apart, and then your reaction rate just crashes. Okay, so temperature, it's really important that we maintain our internal temperature at a point that's optimal for enzyme activity. So we're a warm-blooded animal, so we're able to keep our temperature body constant through homeostatic mechanisms. But other cold-blooded animals, things like snakes, and their temperature changes with the temperature of the external environment. So they have to be very careful, to stay out where it's warm. For example, you always see snakes laying out in the sun because their enzyme activity depends on their environmental temperature. Okay. So a second thing that affects enzyme activity is pH. So most enzymes work best at a neutral pH, a pH of 7. But that's not true for every enzyme. So you can see on the graph over here, pepsin, this is an enzyme in your stomach that breaks down proteins, and you can see it works really well at an acidic pH, and that makes sense because your stomach is very acidic. Arginase is an enzyme that works better at a basic pH, but most enzymes work best at a neutral pH. And really what happens, the reason that most enzymes become denatured if you put them at the wrong pH level is because if you add a bunch of H plus groups, so if you're very acidic, or you take away too many H pluses, those H pluses will interact with the R groups on the enzyme, on the amino acids on the enzyme, and it will cause it to denature or change shape. So then, the last one we're going to talk about is concentration of substrate. And concentration, for our purposes, is just going to be amount. Okay? So you kind of think about it, if you have a lot of substrate, in theory, your reaction will go faster because you can have more enzymes working at the same time. And if you have very little substrate, it's going to be a slow reaction because there's not a lot of substrate for the enzymes to work with. Okay, so how much substrate is available can determine how quickly the enzyme will work. However, at some point, if you look at this graph, you have more substrate, the reaction goes faster, but at some point it does level out. And this is because at some point, all of your enzymes are working at the same time. And if they're all working, adding more substrate won't do anything. So kind of a way to help you understand this is I use the monkey and banana example. Okay, so say you have 100 monkeys in a room, and if you keep giving them bananas, in theory, the bananas will be eaten faster and faster. So if you have 100 monkeys, you give them one banana, the rate of banana eating is pretty slow. If there are 2 bananas in the room, 2 different monkeys can eat at the same time, so the rate of banana eating is faster. If you add 50 bananas, the rate is much faster because now 50 monkeys can be eating at the same time. But if you put 100 bananas in the room, now you have your maximum rate because 100 monkeys can be eating bananas at the same time. But if I put 150 bananas in the room, I'm going to have the same rate as if I had 100 bananas because only 100 monkeys can eat at the same time. So at some point, adding more substrate doesn't give you a faster rate because there's not enough enzymes to actually physically work with those substrates. And finally, I have a couple of things that can influence rate of reaction that are a little different than temperature, pH, concentration, etc. So the first one is the presence of cofactors. So cofactors are basically other molecules that help enzymes to have the perfect shape. So they hang around and they help enzymes to have the right shape. So there are 2 types of cofactors. There are non-protein cofactors. So these are things like ions that help enzymes to fold properly. And then there are coenzymes, which are things like vitamins. So basically, the only reason you have to eat vitamins in your diet, or not the only reason, one of the main reasons you have to eat vitamins in your diet is that vitamins help your enzymes to fold properly and therefore have the proper shape and the proper active site, etc. And then finally, something that can actually slow down the rate of enzyme reaction are inhibitors. So inhibitors are things that interfere with enzyme activity. And there are 2 main types of inhibitors. There are competitive inhibitors. So these are things that will bind to the active site. Okay, so you can see I have a picture over here. So this is my substrate that's supposed to bind to the active site so I can do my reaction. But a competitive inhibitor is something else that binds in the active site that's not supposed to be there and makes it so the substrate can't bind. So this would obviously slow down rate of reaction. And a non-competitive inhibitor is something that binds somewhere else other than the active site on the enzyme. And by binding there, it changes the active site shape so that the substrate can't bind. So that's it. That's everything about enzymes, and I will see you in our last video of the unit, video 8.